All right, and we are back from what seemed like a very quick break. Let me know that you guys can still see and hear me. Hey, Trung, Shima, Dory, awesome, awesome, awesome. If we have anyone new that's just joining us for this session, my name is Kristen Russell, and behind the scenes we have two of my very favorite other teachers, Paul and Andrew. So make sure you hit them up in the Q&A. You can just drop them a hello. You can ask them questions about this session. If you missed anything from last session, you can still ask them about that there. But, you know, we're here to answer all your questions, so we want to make sure we can do that. Hey, Cassandra and Leslie. So glad to see you both. All right, so have all of you guys attended Molecular Genetics Part 1? I won't kick you out if you haven't. I'm just curious. All right, so most of us have, a few of us have not. That's cool. So in Molecular Genetics Part 1, what was like the main topic for those of you guys who did attend? Yeah, it was how we get DNA, right? So synthesizing DNA and going through the process of transcription. Okay, so where do we kind of stop with transcription? Where do we leave off? Yeah, we've produced mRNA. Okay, so we synthesize DNA, we talk about all of our different enzymes that are involved in that process. Then we go through the process of transcription, where we're going from DNA to RNA. Well, now what? Now what? Yeah, now we have to translate these beasts. We need protein out of this. So we're going to use that RNA and we're going to translate it in order to make a protein. So today's lesson is all about translation. So we're going to talk about codons because codons are pretty cool. And we are also going to talk about how we end up with that polypeptide, just another name for a protein. We will also then talk about the 4 levels of protein structure. I'm sure you guys know this stuff pretty well, but we will do a quick review of that. So before we dive into kind of the process of translation, let's talk a little bit about codons. What is a codon? What is a codon? Yeah, so it's a triplet. It's 3 nucleotides, or you could think of it as 3 base pairs, okay? So 3 of those are called a codon. Now, Shima, we can have start and we can have stop. And what else can we have? Yeah, we can have something that simply codes for a specific amino acid. Now, you guys know that on this new MCAT, you have to know quite a bit about amino acids. Do you think you have to memorize the codon for every single one? Thank goodness you do not. That would be pretty awful if you had to remember all of them, okay? But we don't, thank goodness. Which ones do we have to know? Which ones do we have to know? Yeah, we're gonna want to know our stop for sure. Okay, so our stop codons are UAA, UAG, and UGA. Does anyone know a great mnemonic to remember these? I actually do have one for this. All right, yeah, so you guys are basically on the same page with me. We have you go away, you are away, and you are gone. So I'll put that up here. You go away, you are away, and you are gone. Okay? So UGA, UAA, and UAG. That's how we remember our 3 stops. Okay, so what's the deal here? I look at these and I'm like, all right, there's 2 codons that code for phenylalanine. There are 6 that are for leucine. There's 3 for isoleucine. What's the deal? Yeah, we can call this degeneracy or redundancy. So basically what this means is there's going to be multiple codons that code for the same thing. Now, I like what Shima's saying down here. We've got third base wobble. What does that mean? What does that mean? What is third base wobble? Yeah, you can have a substitution in the third base, and most of the time it's going to be fine. Now, if you start to have switches, if you have a mutation in the first or the second, you're likely going to be actually changing the entire amino acid that's being coded for. So I'm trying to think. I think it's the actual genetics lesson where we talk about individual mutations, where you talk about, point versus frame shift and all of that. But basically what we're seeing is if we have a switch in the third base of the codon, it's usually going to be okay. And what type of mutation we refer to that as? Yeah, that one's going to be silent, absolutely. And it would probably have to be a point mutation because frame shift is never going to work out where that's just like makes everything fine in the whole sequence. Okay, so our degeneracy, we said that that means like, okay, we have lots of same codons that are going to code for the same amino acid. There are 20 amino acids that you're going to have to know for test state. Again, you do not have to know all the codons that code for them. You need to know the 3 stop codons, and you need to know start. What is start? Yeah, start is methionine AUG. Okay, very nice. All right, so when we are talking about amino acids, yeah, we have to kind of remember them. All right, so there's 20 to remember. What do I actually need to know about them? What do I need to know about them? Yeah, I want to pay attention to like categorizing them. So it's a lot easier to chunk the data, as we just talked about with our memory and encoding, than it is to remember each one individually, okay? So the first thing is to recognize what piece of this you're looking at when you're talking about a side chain, all right? So you're going to have your carboxy, you're going to have your amino terminus, and you're going to have your R group, and the R group is the side chain that gives the amino acid its characteristics. So you guys are saying, all right, I care about the side chain. I might look, is it polar or non-polar? Is it hydrophilic or hydrophobic? What else might I look at? Is it acidic or basic? Absolutely. Is it aromatic? Awesome. So lots of good things to consider. Now, in terms of the naming of amino acids, what do you have to know about the naming? What do you need to know? You need to know the name, you need to know the 3-letter abbreviation, and you need to know the 1-letter abbreviation. So I know it seems like a lot, but just got to get it done. You guys can do it. I have a lot of faith in your ability to handle that. Okay, I do have a couple of mnemonics that I think are pretty helpful here. So how would I remember all of the hydrophobic amino acids? You guys remember this? Does anyone know this one? All right, so I use family videos were PG. Doggies. Family videos were PG. All right, so again, this is our hydrophobic amino acids. So I want you guys to list these out for me. What is F? Yeah, totally the mailman. I told you when the bell rings, they go crazy. They've been conditioned. Yeah, okay, so phenylalanine. What about A? alanine, okay? M. Methionine. I love the ones that make sense. Some of them seem kind of odd, but methionine. All right, what about I? Isoleucine, very good. And L? Leucine. And the Y is nothing. That's why I have it just lowercase here. And then what about V? Or sorry, W. No, V. I was right. Yeah, valine, good. Okay. W. Tryptophan. P. Good old proline. And finally, G is glycine. Okay, so family videos were PG, gives you all of your hydrophobic. Okay? Now, what about those that are aromatics? You have a good one for that? Yeah, HTTP. I love it. Okay? So HTTP, these are aromatic. So what's H? Histidine. Yeah, and this one, you got to be careful with some of these mnemonics because, like, the first one I gave you went by their one-letter code. This is just a mnemonic to, like, remind you of ones that, like, start with this first letter. So we kind of go away from our one-letter naming here, and these T's, what would they represent? Like, what are the other aromatics? Yeah, tyrosine and tryptophan. And what is actually the one letter for tyrosine? Yeah, tryptophan is W and tyrosine is Y. So you got to be careful with the mnemonics and know what you're doing with them, but it's pretty helpful. Okay, and then what is our P, which is actually an F if we were talking about the one-letter code? Yeah, phenylalanine. So got to be careful with them so you don't confuse yourself with the one-letter code, but these are just two of the mnemonics I like to try and help group these together. So again, you know, big picture here is that we're trying to classify them based on being acidic or basic, based on hydrophobic or hydrophilic. We just want to group them together so that we know something about them, right? And we don't memorize one at a time. Yeah, Shima, I sure can. Let me pull that up again. All right, there you go. If you guys want to take a quick screenshot of this, you are welcome to do that. Give you guys a couple seconds. All right, cool. So let's try our hand at a question now. Let me bring up our poll. And I will pause for about 15 seconds to let you guys get your answers in. Let's see if we can get a pink star out of this one. All right, come on back and get those answers in for me. Now we'll close it up here in 3, 2, and 1. All right, so I'm not going to broadcast yet. I want to kind of talk through these. Now, tell me, when you see a question like this, which of the following is true? Are we going to be making a specific or a general prediction? Yeah, we got to go general, Dory, because we're just going to have to evaluate each of these. Okay, so looking at answer choice A, there are more amino acids than codons. Well, tell me, how many amino acids are there that we have to know about? 20. How many codons do we have? 64. There are 61, including starts and amino acid, and then there are 3 that are stops. Okay, so there are more amino acids than codons. No, that's not going to work. So we can get rid of answer choice A here. I just got to get my line going through the right piece. Okay, let's look at B. Any change in the nucleotide sequence of a codon must result in a new amino acid. We notice this is kind of extreme, and what is this saying? Yeah, there are wobble rules. We talked about degeneracy. So this is saying any change in any nucleotide. So if the codon changes at all, that must result in a change in the new amino acid. And we know that not to be true. Okay, so we can get rid of answer choice B as well. Okay, well, what about C? The genetic code varies from species to species. Yeah, all species are using the same code, so we can get rid of this one, which is going to leave us here with answer choice D. I will broadcast and show you that 75% of us got this one. Very, very nice. So it's true that there is no codon that can code for more than one amino acid. We saw that there are multiple codons that can code for the same amino acid, but it doesn't go the other way. Awesome, awesome. All right, let's take a look at another one. I bet we can get a pink star on this one. This one's pretty quick, so I'll give you like 5 seconds. All right. Ooh, I like it. Paul bringing in info from our last lesson on memory. Let's retrieve that information. All right, go ahead and get your answers in. And I will close it here in 3, 2, and 1. All right, so codons that specify the same amino acid can differ only in the. Can we make a specific prediction here? Oh yeah, we can. We can make a specific prediction because the wobble effect occurs in the 3rd base, okay? So the correct answer here is C. So about 72% of us got this. I want to go back just briefly to our first image so you guys can see this again because this is important that you understand this. So let me see if I can find the right slide again. Here we go. All right, so what we're saying with this thing is if we look at UUU versus UUC, so we're right up here. So the first U would be the first one, the second U would be our second one, and then the third, where it's U versus C, would be our third. So based on degeneracy, we're seeing that the 3rd nucleotide can change, but it will still produce the same amino acid, okay? So 3rd one can change, which you can see all throughout this chart. First and second, not so much. So our correct answer here, as the majority of us said, but not quite all, is answer choice C. Okay? Austin says, question doesn't assume stop, though. Yeah, so this one says coding for the same amino acid, right? Yeah, that could have been tricky, I agree. Okay, so in our next learning goal, now that we've talked a little bit about amino acids, which I know is your guys' favorite topic, we're going to actually talk about translation. So now we're going from the beginning, what we said, we have an RNA that we produced after transcription, and now we want to produce a protein. Okay, so what is kind of the first thing that has to happen? What has got to happen here? All right, so where did we leave off? Let's figure out where translation's beginning, like what has to happen. All right, I like it. We have to go to the cytoplasm first because where was the mRNA when we left off in transcription? Yeah, in the nucleus, okay? So the mRNA is going to go through nuclear pores in the nuclear membrane to head out into the cytoplasm. All right, then what? Yeah, so we're gonna care about the ribosome now. So all the mRNA is doing is messengering it because M is messenger. So it's messengering the genetic code into the cytoplasm so it can find a ribosome. Now, what is a ribosome? What is a ribosome? It has protein and what else? Its function is to assemble proteins. What is it made up of, though? All right, it has subunits. This is true. But its two pieces that like compose the ribosome are rRNA, so ribosomal RNA, and protein, okay? Those are the two things that actually make up a ribosome. Now the function, as you guys were very accurately saying here, is the site and organelle, or I guess we don't really consider it an organelle here, but piece of the cell. The site and the piece of the cell, or the functioner, that is going to be able to produce proteins or carry out protein synthesis. All right, so you guys mentioned subunits. What do we need to know about subunits? It is different between eukaryotes and prokaryotes, quite true. Now we're going to have big and small. So what do we have in eukaryotes? What do we have in eukaryotes? Yeah, so we're going to have the 80S overall, but we're going to have the 40 and 60. And so what is the S representing? What is the S representing? Yeah, it's just a unit of size, all right? That's just telling us how long it is. So how many pieces are making it up. Okay, what about prokaryotes? Yeah, prokaryotes are going to have the 50 and the 30, so overall we would call it 70S ribosome for prokaryotes, and eukaryotes are the 80S. Okay, so what's the whole point of these subunits? Like, what's going on here? Yeah, I gotta have both pieces of this to make a functional ribosome. So the ribosome is actually going to attach itself to the mRNA. All right, so why does the mRNA need to be attached there? Yeah, so that it can be read. And what else does the ribosome do? Yeah, it's like the site that tRNA wants to go to. So not only does it hold the mRNA in place, it's like holding everything. It's saying, okay, you come over here, and I'm going to hold you here while you create my new protein. So it's actually stabilizing this entire interaction that we have going on. So tell me this. If I have one sequence of mRNA, am I only going to have one ribosome? Ooh, I like that, Dory. The usher of translation. Love it. Yeah, and you guys are correct. I can have several ribosomes attached to a single piece of mRNA. Polyribosomes. Awesome. All right, now what's the deal with tRNA? What's the deal? What's the T? Yeah, transfer. So what we're doing here is bringing something down. So how long is the transfer RNA? You guys know? So tRNA has about 75 nucleotides, but 3 of them are referred to as our anticodon. So it's not just 3, it's longer than that, but we have an important piece that's going to be the part of interest. So the tRNA is going to actually read the code, and it's going to bring the amino acid that we want. It's 75 total, and then 3 of those 75 are the part we actually care about, okay? So if you look at this picture right here, this is a good example, right? This entire structure is your tRNA, and here is our anticodon sequence, the important part, right? Okay, so again, we are looking for a codon match. So our codon in our mRNA is going to match with an anticodon on our tRNA, and that's basically bringing on our amino acid, right? So each codon is going to be read by one tRNA or multiple. How does that work? All right, so does one tRNA have multiple amino acids on it? No. So how does that work? Is each codon going to be read by one tRNA or multiple? Yeah, it's got to be multiple, right? So this tRNA is going to swing by and it's going to read the codon. If it recognizes a match between the codon on the mRNA and its own anticodon, then we will get the amino acid. But if it doesn't match, it's going to keep on rolling and another tRNA is going to come through. That's how we're bringing amino acids to the mRNA to make a fully functional protein in the end. Does that make sense? We feeling it? All right, cool. So let's add some more complexity here because we got to make it exciting, right? So let's talk about the mechanism of this actually occurring. So there are a few different sites that we need to be familiar with when we're talking about translation. What are those sites? There are 3. Yeah, so we have the P site. What's the P site? What's the P site? Let me tell you what page this is on so you guys can go back and look at this too. It's on page 229, and you can see the picture there. I actually like the picture in the book a little bit better than the one on this slide, so I would just kind of like make some notes in reference because I think the one in the book has it better laid out where you walk through the steps. This one we're getting kind of a cyclical structure. It's on page 228 and 229, okay? All right, so 229 talks about A site, P site, and E site as well. So what is the A site? What does it stand for? That kind of might help you a little bit. Yeah, A is our amino acyl. P is, I'm looking in the book to make sure it's in there. Yeah, it is. So it goes APE in the book, but peptidyl transferase. And then our E site is kind of our ending, all right? So our small subunit of our ribosome is going to kind of start ahead of where we actually want to start going through and sequencing this thing. So which direction is it going to move on the mRNA? Five prime to three prime, right? That's always the direction that we like. Okay, so it's going to start moving, and when is it going to actually do something? Yeah, it's got to find a start. So the start codon again is AUG. All right. So what do we call the space between where it hops on and where it starts? What do we call that area? Where the ribosomal subunit attaches and where it actually begins to translate. Absolutely, Alex. We call that the 5' untranslated region, okay? Now, once it gets to the start codon, what else happens? What needs to happen now? It encounters a start, and then what happens? We can look at our picture up here, and we see, okay, 5' to 3', we're going along. Oh, hey now, I see a start. Well, right now our ribosomal subunit is the only thing that's bound to our mRNA. So what else do we need to actually do this? Yeah, Trung, we need our large subunit, okay? So we know that methionine is our start amino acid. We know that in order to actually translate something, we need our large ribosomal subunit as well. So what site do we care about here? What site do we care about? The P site. Right now we care about the P site. So we are joining the large ribosomal subunit to the small ribosomal subunit, and we need one other important thing. What is our tRNA that carries that methionine called? What do we call that? The tRNA that carries methionine. Well, it's beginning everything, right? So we could refer to it as the initiator tRNA. Very nice, Austin. Okay, so the initiator tRNA is going to bind to the P site, okay? So this is where we start out. P site is where initiator tRNA is going to bring in. We get our methionine added. Now we have both our large and our small ribosomal subunits. Okay, now things are getting a little more complicated, right? Okay, what happens next? The picture's getting more significant. What happens next? So our initiator went directly to the P site. So we've got one amino acid added, the methionine. So now we're going to have our aminoacyl tRNA. So this is like the non-initiators. These are the ones that have just a regular amino acid attached to them. They're going to come down. They're going to take a look at the next sequence. And where are they going to bind? Where are they going to bind? Yeah, they're coming to the A site, right? Makes sense. Aminoacyl tRNA binds at the A site. So we initiated in our first step, and now we're elongating because we're scanning here to see if we have a codon-anticodon match. We're bringing that amino acid in. Now, what else do we need other than our aminoacyl tRNA, our A site, our large and our small ribosomal subunits? What else do we need? We need energy. Our source of energy here, as Austin says, is GTP. What else do we need? EF, absolutely, Shima. EF is elongation factor, okay? So elongation factor, GTP, our aminoacyl tRNA, now we're in business. Okay, so the methionine, which was our start one, is covalently linked to the incoming amino acid through what type of bond? What's going to link amino acids? Peptide bond, bingo. Okay, now once we have that bond, what happens to the initiator tRNA? See ya. It's gone. All right, so once we have that bond formed, the initiator tRNA is going to be let go from the P site. Now what happens to the ribosome? Now what happens to the ribosome? It's going to move forward, right? Ribosome's still there because we got more work to do, but it's going to move down one, okay? Okay, so we've gotten rid of the initiator, and we originally had the amino acyl tRNA attached to the A site. But now we're moving down one, so what needs to happen? So what needs to happen now? Well, remember, we said all of the amino acyl tRNAs are originally going to be binding to the A site. So if we want to add more, what do we have to do to the one that is currently there? We got to move him over. We have to move him to the P site. So the P site is going to be the initial site of binding only for the initiator tRNA. However, we can move from the from the A site to the P site in order to make space on the A site for a new amino acyl tRNA, okay? So we're going to shift everything over, and now we have new space. So this is how we're going to keep elongating this entire thing. All right, so we're adding new pieces, we're shifting things over, our ribosomal subunit shifts over by one. Let me know if you guys are following so far. Again, I like the picture in the book a little bit better. It's easier to see the individual sequences. All right, so just a quick recap of what we saw here. So we have our initiator tRNA with methionine attaching at the P site. So from there, we're going to bring on an amino acyl tRNA at the A site. The amino acids are going to form a peptide bond. The initiator tRNA is going to bounce off the P site. Now the A tRNA and the amino acid at the A site can shift to the P site, freeing up space on the A site for another tRNA. So we keep going. We're growing this chain using GTP and elongation factor. Then what? Then what? Yeah, eventually we're going to have termination, and we come to termination when we reach our stop codon. Remember, what were those 3 stop codons? What are those 3 stop codons? UAA, UGA, and UAG. Very good. Okay, now do we actually have an amino acid attached to the tRNA that matches the anticodon for the stop codon? No, right? There's nothing there. So what is actually going to tell us to let go when that tRNA reaches the A site? Yeah, termination factor, or we can also refer to that as release factor, okay? So yeah, and Paul's mind is absolutely the way to think of this. So I didn't clearly say before that when it's time to peace out and get out of there or let go, it's going out through the E site. So once that peptide bond is formed between the 2 amino acids, things are shifting P site to E site. E site allows it to exit or take off. All right, let me know if you guys are having a solid understanding of these different pieces. I think this is the part that maybe for students coming in who think they have a really strong understanding of translation, this is probably the hardest piece. So if we got this, then we're going to be solid. All right, perfect. Then the rest of this is just going to be like easy, right? We got the rest of this, no problem. Okay, so post-translational processing. Once we've actually done our whole shindig, what else do we have to do before we are totally done? Yeah, so we're like, we've now made our primary structure. So primary structure, which we'll talk about in a little bit, is just our sequence. So we have that down, but we need to now make the protein functional. So we're going to add some molecules that might help us with our activity. We're going to be able to add things that help the protein withstand the places in the body that they're going. We're going to see different modifications. What kind of modifications are we going to see in terms of adding these molecules on? How's that going to work? All right, I like what I'm seeing here. So again, the reason we're doing this is to make it withstand the environment and make it function correctly. So we're going to have covalent modifications through peptide bonds at the N-terminus, the C-terminus, and amino acid residues. And we're going to have non-covalent modifications, which would be like adding cofactors, folding this bad boy up, or allowing us for trafficking, okay? So this is all happening after we have stopped, after we have released, and we're good to go. And now we're just making our functional protein. All right, let's try a quick question to make sure we've got it. So I will give you guys about 20 seconds to assess the question, plan, execute, and answer. All right, get those answers in, and I will close it up here in 3, 2, and 1. All right, it looks like about 95% of us are on the same page here with answer choice B. That is correct. Pink star time. Fantastic work on this one. Yeah, go you guys. You're awesome. Which of the following occurs when the eukaryotic translational machinery encounters the UAG? Absolutely. This is a stop codon, and we said when we hit a stop codon, we need the termination or release factors in order to end translation. Awesome job there. All right, let's try another one. You guys are killing it. Let's see if we can get another pink star. About 15 seconds on the clock for this one. All right, go ahead and get those answers in. And we will close it up here in 3, 2, 1. All right, it looks like about 70% of us were coming in with answer choice C. Let's walk through this. All right, a certain cell is exposed to a chemical that inhibits ribosome movement. So the first thing we would ask ourselves is, what do ribosomes do? What are they involved in? Yeah, so we would think our strategy here would be like ribosome, we think translation. Now, if we had issues with the ribosome, would that have any like hindrance on DNA synthesis or transcription? Which one comes first? Which one comes first? Yeah, remember, we get DNA synthesis, then we get transcription, then we get translation. Okay? So if we had a messed up ribosome, we'd still have transcription functioning normally. So what answer choices can I now get rid of? Yeah, A and B are goners. Woo. All right, A and B are gone. So now I look at C and D. Prevent effective protein synthesis or decrease speed. Now, in order to synthesize a protein, what does the what does the ribosome have to be able to do? It has to move. We talked about the importance of the subunits being able to shift along the mRNA as they're moving. They have to be able to move. So if they cannot, we're not going to be able to effectively produce a protein. Nicely done. All right, so last learning goal here is to talk about protein structure. You guys probably have seen this 1,000 times in school, so I'm going to do a lot of asking you to tell me what's going on. So what is primary structure? We talked about it a little bit already. Yeah, this is just the peptide bonds that hold together the amino acid sequence, so it's just our polypeptide chain. Now, we said our peptide bonds are covalent bonds. This is made in our process of translation. We have both an N-terminus and a C-terminus to this structure. All right, what about our secondary structure? What about our secondary structure? Yeah, this is where we see our alpha helices and beta pleated sheets. So we would think of these as like substructures, so they're somewhat localized. The two main types are the ones we just mentioned. So we're going to see interactions here. And what type of bonding are we going to see? H bonds, absolutely. So we're going to see H bonding on that peptide backbone that creates the beta pleated sheets or alpha helices. Okay, so that's our kind of substructure. And then we have tertiary. What's tertiary? Now we get our overall structure, so our 3D structure of our entire protein. So this is where we take like the alpha helices and beta pleated sheets and actually kind of like fold them up all together into our final shape. So what drives this folding? Yeah, so we're going to have hydrophobic, non-specific hydrophobic interactions that are going to allow folding. But does that stabilize the final structure? That just allows us to move around. What actually stabilizes that final structure? What gives specificity? Yeah, okay, so salt bridges, hydrogen bonds, and like tight packing of our side chains through disulfide bonds, that's what's going to give the specific interactions. So non-specific hydrophobic lets this guy fold together based on, what R groups it has and all of that. And then we're going to create specific interactions to like lock in the final 3D structure using salt bridges H bonds. Okay? Yeah, still H bonds involved. All right, and then finally we have quaternary. What's that? Yeah, this is where we get multiple subunits. So do proteins have to have quaternary structure? No. Do they have to have primary, secondary, and tertiary? Yes. They must have the first three. They do not have to have quaternary. So what's like an example that we think of as probably the most commonly used example for quaternary structure? Yeah, hemoglobin, all about it, absolutely. Okay, so we talked about the main ideas of protein structure. Now, when we're looking at this thing, we can build it up. We can also break it down. So what can cause denaturation of a protein? Heat, pH, there's lots of things that it can do. How is it actually breaking it down? What level of structure is denaturation breaking down? Yeah, so if it has quaternary, it's breaking that down. And it's also breaking down secondary and tertiary. because what we're seeing now is just one long chain of amino acids. So it only has its linear structure remaining. So that's denaturation. So we can also have hydro interactions now. So if we have hydrophobic and hydrophilic areas, what part wants to be on the outside? Yeah, because there's so much water on the outside, all the hydrophobic pieces are going to want to cram to the middle, and all the hydrophilic pieces are going to want to be on the outside. So this kind of demonstrates how that initial 3D folding is happening just due to hydrophilic and hydrophobic regions. All right, let's try another question. I will clear out our poll and give you guys, this question's a little longer, about 25 seconds. Go ahead. All right, go ahead and get your answers in. Okay, yeah, so hydrophobic bonds, that terminology may confuse you. Think of bonds as the same thing as interactions. So if you want to change your answer, think of like these things as interactions. If you want to change your answer, you can do that now. And I will close it up in 3, 2, and 1. All right, so the majority here is saying answer choice B, even though we had kind of a weird term. So several types of non-covalent bonds are involved in protein-protein interactions and by extension, interactions between portions of a single protein. Which of the following bonds are apt to be more common in the non-aqueous interior environment of a protein than in the aqueous surface environment? Okay, so non-covalent, so we could get rid of something pretty quickly here. What can we get rid of? Yeah, get rid of answer choice D, right? That was like silly that they even put that in there. So D is gone. All right, well, it tells us that it's non-aqueous. So what else can we get rid of? Yeah, we're probably not going to see our H bonding like super prevalent in here. And does ionic make sense? No. The only thing we're going to have, and which totally makes sense based on the idea that we're non-aqueous and we want to be on the interior like we saw in the last image, would be our hydrophobic interactions or hydrophobic bonds. Okay? All right, let's try another one. You guys are rocking these. I want to see another pink star before we're done. All right, so I'm going to pause for about 15 seconds and let you get your answers in on number 11. All right, get those answers in, and I'm going to close it up in 3, 2, and 1. All right, our majority here putting answer choice A. Which of the following statements about protein denaturation is correct? And again, we have to use elimination. So we look at answer choice A. Denaturation can be irreversible. All right, can be, that's nice moderating language. We may not know that for sure, but it sounds like it could be possible, so we'll save it. Let's look at our others. B, proteins are pH and temperature resistant. What do you think about that? No, which is why they have certain environments they like to be in, right? If proteins were temperature resistant, it wouldn't be dangerous for us to have fevers. Having really high fevers like 105, doctors start to worry about your proteins denaturing. All right, denaturation only occurs through enzymatic modification. That's extreme, absolutely. Get rid of that beast. It's out of there. And then finally, D, which looks kind of weird here in the way this is laid out with A, B, C, D. Denaturation does not change the tertiary structure. Now, we already said it definitely does change the tertiary structure, so that one's out of there, and now we can go back and see answer choice A is absolutely correct. With that, we have finished our molecular genetics part 2 lesson on translation. I would love to see you guys in my next hour, which is going to be focused on blood. So take a quick break, and we will see you back here in just a couple of minutes.